Hi, good morning. My name is Araceli Naranjo, and I am here representing the GMP manufacturing side. So I have interacted with a lot of the people that are here in the room, and just want to kind of give you guys the other end of the CAR T therapy side. So first, I want to introduce our team. Our team is pretty large. Everybody that comes through our team is trained in order to be able to make these manufactured T cells for our program here. So I'm very proud of everybody here. And I know you guys have seen this picture again and again, but basically talking about how we take a product from the leukapheresis. We then isolate the cells of interest. Each of these IRBs and INDs that we deal with actually have different kinds of cells that are of interest. So we take those cells of interest, we then genetically modify them with this CAR T lentivirus. We then expand them outside the body in our GMP facility here on campus. And then eventually they get prepared and infused into the patient, whether like they spoke about before, ICV, IV, or yeah. These are some of the protocols that we're currently working. A lot of you guys are very familiar with these. Whether we're targeting for CD19, CD33, CS1, IL-13 receptor alpha 2, HER2, chlorotoxin, PSCA. And our production facility is actually here. It's called the CTPC, Cellular Therapy Production Center. It's across just north of the Helford Center. We're located on the 4th floor of Beckman. It's a very small area. You can kind of see in the bottom right is a quick scheme of our facility. Within our facility, we have 6 production suites. Of those, we house 4. So that's how much work we have. It's a clean room, so it's ISO 7. That means it's 10,000 class. So when we go in, we actually have to gown up, proper PPE. We do not wear street clothes. We wear scrubs, hair. We cover our hair, eye shield, face mask, gloves, disposable gowns, shoe covers for our shoes. Our facility was actually established here in 2010. I've been here since the late 1990s. So I was actually been a part of the growing of this program. So I'm very proud to say that we've been expanding, moving forward. And before we used to work in a tiny little lab, and now we have this really awesome GMP facility suite. What you see here is very basic with our biosafety cabinets, our incubators, a lot of very intricate devices, very specialized for what we need, what we do. In a room like this, we can actually have 2 teams of 2 working. Whenever we work, we have to work in teams of 2. Kind of like I hear a lot of like checks and balances that are done here. We're the same. We have a manufacturer. So that person's actually the one manufacturing. We have a verifier who's reading the standard operating procedures, and they go step by step. And they also are the verifiers reading the procedure as well as verifying that the manufacturer is doing what the SOP says. If there's any deviations, we go ahead and mark those down. But yes, we work in teams of 2. One can work on each side. Each team cannot work on more than 1 product at a time. So we ensure that there is no cross-contamination of any kind. Not just is it just the teams, but also we segregate reagents, materials, devices, so there is no crossing of any kind. We deal very closely with the Donor Apheresis Center, which I'm sure everybody knows is in the Amini Center on the 2nd floor here on campus. So what ends up happening, I know that there's a lot of stuff that is done from the clinical side with Julie and teams. At this point, when the patient comes in for a leukapheresis, it's usually about a 4-hour blood draw. However, there are complications. And so we deal very closely with the Donor Apheresis Center to be able to tell us kind of what time to pick up. Once we get the green light, we come over to the Amini Center, walk on over with our transport container, and then bring it over to our GMP facility where we deal with our quality assurance team to ensure that the product is for this intended patient. We then are able to take it inside our GMP manufacturing suite. Like I said, I've been in this program since the late 1900s, and a lot of these devices weren't available back then. And so it's really awesome to see the progress that we've made, not just in the amount of protocols that have been opened and the different, medications that you guys are able to give, but also in the devices that we can use. A lot of the times we didn't have these things, so we were doing them manually. The first one is called CPAX. You guys might be familiar with it. This, for us, we are able to isolate the white blood cells, the initial separation. And then we have a Clinimax device. This one's a Clinimax Plus. This is from Miltenyi. They make different types. This is actually my favorite device. It's been around a long time. It's like a workhorse, but this is really important because, again, it helps us isolate the cells of interest. So again, for every protocol, we're looking at different cells, whether it's central memory, T-naive memory, or just some depleted cells. This is a device that allows us to do that. Next is an incubator. This is where we house our cells. This is where they live for the most part. We take them out, we work with them, we put them back in. These have a certain humidity, temperature. I think someone mentioned that there's these beads that we use in order to be able to stimulate the cells, CD3, CD28 receptor. Well, those beads can go into the patients. So part of the criteria is that we remove those beads. And this, what's called a magnetic particle concentrator, is actually a huge magnet. And we basically put our cells on there in these bags that allows those beads to adhere to the magnet, and then we flow the cells out. We do that more than once because obviously if we fail that as a criteria, we can't do anything with these cells and then they fail. So we make sure that our team is very aware. We do a lot of visual observation using microscopes, and if they see one bead, we actually have to repeat this whole process. The Lovo device is another device that has been, I would say, more recently implemented, but in the past, we would have to do centrifugation of cells in order to wash out our cells from the media that we have them. And because they have a fetal bovine serum in them, obviously that cannot go into the patient, so we do very thorough washing. This Lovo device allows us to do a process that used to take us anywhere from 4 hours down to 45 minutes. So as you can see, it really helps us out. The next device that we encounter is a control rate freezer. It does what it says. It controls the rate of the freeze. That ensures that the cells are as healthy as possible so that when they are being frozen in the liquid nitrogen, by the time that they come to you, whether it's in 4 to 6 weeks, in a month, in 6 months, that they're healthy and they're safe for the patient. There's other devices like the Muse-Cell Analyzer where we count the cells. So it's not just visual inspection. Obviously, you guys need a certain dose. So that Muse allows us to count while we're expanding the cells, as well as in order to get the number that you guys need to prescribe to the patient. Then there's a Walders, which you guys are all aware of, sealers, and a pump. So these are all the things that allow us to do what we call a semi-closed system and the expansion of our T cells outside the body. This is a really cool picture just because you're able to see kind of what's going on. I know you guys see this firsthand in patients, but this is what we would see in a flask. So what we did is we grew some tumor cells, we exposed them to our CAR T cells. Through time, you can see that they apoptosis, and then those T cells are no longer present in the last picture because they are actually off to the next target. So they're actually just continually helping each other out. This is what the expansion bags themselves look like. So when we talk about expanding of the cells, they are in these bags. They're very beautiful, clear bags. We're able to see them, put them on a microscope and actually see what they look like. So it's actually really nice to see the process as they're going through. Depending on how many cells we get, and I know that, again, it's been touched on the quality of the cells that we get. Obviously, if the patient's not doing great, we will get a product that's not great to start with. But our team does their best in order to expand these cells outside the body. So we really care for them. We start in very small bags, like as you see at the very top. These are called 32 View Life bags. They're probably no bigger than this, a little like 3x2. And then we go to bigger, and then that big one that you see actually holds 2 liters worth of T cells. We are very close at monitoring, not just like the way that they look, but the way they expand, the cytokines that we give them, and continue to expand them outside the body into the doses that we need. Now, we don't just expand to the dose that you need. We also expand. Sometimes we try to do 2 to 3 times because we know that these patients can get multiple infusions. So we want to make sure that we can cover as many as possible. I think for some of our GBM products we've done, I think the requirement is 3 to 4. I think we've done up to 14, 15, 16 infusions. So we're really proud of that. This is what you see. This is what we see when we put them under the microscope. So you can see there's these clustering. So again, those beads that we talked about that help stimulate the cells, this is what it looks like. So in the bottom right, you can see what the bag looks like undisturbed, where those beautiful brown clusters, those are not T cells. Those are actually the clusters of the beads that help them expand outside the body. I know this looks like the moon, but it is not. no eclipse going on here. These are all our T cells. These are beautiful, live, growing, thriving T cells. The expansion bag on the right is what you see, and then the picture on the left is what we look at when we take our pictures under the microscope. Again, these are the things that we do on a daily. Our team is trained. We make sure if we see any oddities that, I'm the supervisor, so I go ahead and communicate with the team on a daily basis. Everything that they see, we talk about, we discuss, make sure that we're doing our best possible. These are some images of our team in action. So rolling out other devices, working together, reading the standard operating procedures, working under the BSC. Obviously, there's no patient products being used here. This was actually done for, I think there was some pamphlets that went out, just kind of want to talk about our GMP work here on campus. Bottom right, again, looking at under the microscope at the cells, as you can see that manufacturers kind of moving the cells. Obviously, when you look at the microscope, you're looking at a pinhole of a small bag, but we do our best to try to look around as much as possible, inspect, make sure there's no particles, make sure there's no gross contamination. Knock on wood, we're pretty good about that. And so yeah, it really does take a powerful team to do this. As you guys know, we need to not just have interaction with you guys, but within ourselves. We need to make sure that we have clear communication and collaboration. I think the next part's really important because we deal with all of these teams all together, right? We have the R&D where this kind of starts. You have the process development where we say, how can we get this from the research side to a GMP space? So that's where we come in. We help with that. We help facilitate that. And then there's a manufacturing. That's what we're doing. And then there's obviously the clinical trials that these products are going to. There's a ton of people that go into this. A lot of the people in this room that work very hard to get these things done. And the clinical trials, what's important and what's awesome about phase 1 clinical trials here at City of Hope is that we can talk. We can have a meeting today, tomorrow, the next day, and discuss what you're seeing, and then go back to the drawing board, right? Go research. And so think about what are the next steps? What can we do better? And all of those things that we research is not just for R&D, but it then progresses to see what we can do in manufacturing. There was one protocol where we were doing a certain type of cell type. And in the literature, it said that another cell type might be more powerful. The team decided in the research side to go ahead and make those cells, put them in mice, and then right then and there, go to the FDA. Within a matter of months, we were manufacturing the cells that they asked for. So that's the powerful thing about being on a campus like this at City of Hope, where we can have those discussions and have the work done all under one roof. And that is it for me. Thank you.